cover chapter 6, cell membrane. Okay, today we will cover chapter 6, cell membrane. Chapter 6 is composed of the 5 subjects, starting from the biological membrane, and also second part is that cell membrane is important for cell addition and recognition, and also we will cover substances can cross the membrane by passive process, and also we will cover active transport. Finally, we will cover the large molecules which enter and leave a cell through a vesicle. Okay, so this is the first subject. Biological membrane are lipid protein bilayer. Okay, so this is, we call it as a fluid mosaic model. Fluid mosaic model is composed of mainly two parts. First one is the phospholipid. So phospholipid forms phospholipid bilayer. Bi means that two layer. One layer, another layer. Okay. Phospholipid forms a bilayer, and also proteins. So this is phospholipid, and there are several proteins is like this. So proteins are embedded, 파묻혀 있는, embedded or associated, means the bind to membrane is like this. Bind to membrane is like this. Okay, so and it means that fluid mosaic model means that membrane is flexible, is not rigid. Fluid mosaic model. Okay, so we will see some details. Okay. Okay, so starting from the phospholipid bilayer. In chapter 3, we cover the saturated fatty acid and unsaturated fatty acid. For example, the saturated fatty acid, there is no double bond. Okay, however, unsaturated fatty acid, their fatty acid, we have the one or more double bonds in carbon chain, so it results in cork. They make this kind of structure. Okay. For example, so in this dimension, there are 1, 2, 3, 4. There are 4 phospholipids is there, but whenever the cork is happen, in same dimension, there is only 2 is available. Okay. So when there are lots of the unsaturated fatty acid, it means that the oil becomes liquid at room temperature, like this. Okay. If we have lots of the saturated fatty acid, they are solid at room temperature. However, if we have lots of the unsaturated fatty acid, they are liquid at room temperature. Okay. Okay. So phospholipid bilayer is that they have the hydrophilic head. They have hydrophilic head, and also they have hydrophobic tails like this. Hydrophobic tails interacting with each other in the inside. Okay. Okay. And also in the phospholipid bilayer, the cholesterol is there. When the cholesterol is there, the phospholipids becomes more flexible. Okay. Okay. So the fluid's mosaic model is composed of the phospholipid bilayer. They have the head part. They have the hydrophobic tails like this. Okay. For example, each phospholipids, they frequently move to here and there like this. But sometimes not easily happen. In this side, there is one phospholipid is there. The other side, another phospholipid is there. This is recorded flip-flop, but this case is not easily happens. But this kind of the rotate or lateral position, this kind of thing is frequently happen, but this kind of flip-flop from one side to another side is happen, but not easily happens. Okay. And then we will cover membrane protein. Okay, membrane protein is composed of 3 proteins. First one is the peripheral membrane protein. So this protein we call it as peripheral membrane protein. Another protein is integral membrane protein like this. Okay. First of all, the integral membrane proteins, you will see that the protein is embedded in the membrane. Okay. That they penetrate membrane, membrane is like this. Okay. However, peripheral membrane proteins, so for example, the protein which is located in the inside, lots of protein, they are hydrophobic. Okay? So if you run the protein engineering course, at that time I teach you that around here, there are lots of the alanine and leucine, isoleucine, but today I will not cover. The important thing is that integral protein, the protein which is located inside, they are very hydrophobic. Okay, but however, peripheral proteins, they are lack hydrophobic region, so they cannot penetrate the membrane. However, integral proteins, they have hydrophobic region, they can penetrate the membrane is like this. Okay. Next is that final one is anchored membrane proteins. Okay. For example, this kind of protein we call it as anchored. Okay. For example, so this is the phospholipid which has 2 fatty acid pair, and they have hydrophilic head. Sometimes the head is several glucose can attach it to the head, and sometimes in the head the polypeptide can be covalently attached to this head. Okay, in this case, we call it as this protein as anchored protein. Anchored protein means that covalently, they have the carboxyl group, peptide has carboxyl group, and between the amine, they have the covalently bonded like this. Okay, so anchored membrane protein is covalently attached to fatty acid. For example, they are covalently attached to this fatty acid. This is we call it as anchored membrane protein. 3 kinds of peripheral, integral, and anchored membrane proteins. And also membranes are constantly changing. For example, the membrane proteins, okay, fluid mosaic model means that membrane is not rigid, they are flexible. And also membrane is constantly changing, especially membrane proteins. Okay. For example, one example is that this mouse cell has the membrane protein. So membrane protein is labeled with green dyes like this. Human cell also has membrane protein. They are labeled with red cells. So we can fuse mouse cell with human cell. So this is a fused cell. At first time, there was a boundary between the green and red protein. But after 40 minutes, you will see that red protein and green protein, they are mixed to each other. So based on this experiment, we can say that membrane proteins can diffuse rapidly in the membrane. So membrane protein can diffuse rapidly in the membrane. Okay? Okay. So last year I give midterm exam, I give some example like this. What was the conclusion? Based on these experiments, what is the conclusion? The conclusion was that membrane protein can diffuse rapidly in the membrane. Okay? Did you understand it? Okay? It means that it is important, okay? Okay, and also fluid mosaic model. At first time, I did not mention fluid mosaic model. They are mainly composed of the phospholipid and also proteins. But you will see that some kind of the carbohydrate is also attached to here. But at that time, I did not cover. But now I will cover the membrane carbohydrate. Okay. So cell membrane also has the carbohydrate. For example, so this is the phospholipid. Okay, some carbohydrate can attach it to this lipid, or some carbohydrate can attach it to this protein. Okay, so this kind of the carbohydrate, we call it as the glycolipid. Carbohydrate which is attached to protein, we call it as a glycoprotein. Okay? So this glycoprotein and glycoprotein, they plays an important role cell recognition. Okay? This lipid, this glycolipid, glycoprotein, say that my name is Ijeon, my name is Kang like this. This is name tag. This is the name tag of the cells. Name tag. Okay. Based on this name tag, some cells can recognize themselves, or some cells recognize others as the enemies, like this. So this glycoprotein and glycolipid is basis for rejection of foreign cells by immune system. Our cell can recognize the foreign cell based on this glycoprotein, glycolipids. So this is the name tag. 제자는 wake up. Okay. Okay. okay? Makeup, okay? 잘 이해하고 있나요? 오케이? 너무 잠이 오나요? 메이크업, 화는 안 자고 있지 맞지? Okay. So, carbohydrate, the glycolipids and glycoproteins are recognition site. Okay. Okay. So now we will go to the second subject. Cell membrane is important in cell addition and recognition. Okay. Previously, I mentioned that glycoprotein and glycolipids plays important role in cell-cell recognition. Okay, so I will show you this experiment. So in the sea, there are lots of the sponge. How do we call in Korean? Sponge? 해면이라고 하죠. 해면 맞죠? So figure 6.5 show that tissue from the red sponge contains the cells like this. Okay? So we can separate each cell by passing it through the fine mesh screen, very fine mesh screen. So we can divide it by each cell like this. We can separate it like this. Okay? But sometimes later, we'll find that identical sponges happens like this. Because if you see here, each cell, they have the protein and carbohydrate. We call it, this is glycoprotein or glycolipid. So protein and carbohydrate at the cell space, they recognize each other. Okay, so they bind to the here like this. Okay, so they will make the same structures like this. So each cell, they recognize, each cell, they recognize, each cell, they recognize, like this. Finally, they will make the same sponges like this. At that time, cell has a protein and carbohydrate. Protein and carbohydrate is their important role in cell addition and recognition. Okay? So sponge cells are easily separated and will come back together again. Okay? Now we will cover 3 cell addition molecules. So we already covered, maybe you guys already know that. First one is tight junction. The second one is desmosome. And final one is gap junction. Okay, let me start from the tight junction. So cell membrane is an important role in cell addition. Okay, at that time, especially the tight junction protein is involved in. Okay, for example, this is one cell and the other cell. Between them, there is a tight junction. Okay. If you see that each one cell is the other cell, between them there is a tight junction is like this. So tight junction, okay, they forms a kilted seal. Kilted. So this is sewing machine. They make it is used to make the kilt. So they will form like the quilted steel is like this. So after they make the quilted steel, they are impermeable junction. For example, when the tight junction is happen like this, or bacterial product or another antigen cannot penetrate this junction. However, the tight junction is impaired. Several bacteria and other antigen can penetrate inside like this. However, tight junction, they make the quilted steel like this, so they become the impermeable like this. Okay? Impermeable. 아니 이제 이해했지? Okay. Next one is we call it as Desmosome. In Korean, we call it as 접착판. It looks like the button. It looks like button like this. In our clothes, sometimes we have this kind of button. So it looks like the button. So based on the cell used the desmosome to link adjacent cell to link together. So it looks like the button. And they link each other. However, this link permit materials to move around them. They are not impermeable. Okay? So this is desmosome. So this is the button like this. So this button is several cytoskeleton bind to this button like this. Okay, this button, one button, the other button is like this. So this is structure we call it as the desmosome. But the important thing is that this is the permeable. Okay. The main difference between this one, tight junction, and desmosome, this is impermeable, but this is permeable. Okay. Final one is the gap junction. Okay, gap junction. This is gap junction. This is the tight junction. This is desmosome, and this is gap junction. Gap junction. So in gap junction, connexin, this channel protein plays important role. The connexin, they bind to each cell and another cell, they bind together by connexin. So connexin has the small pores, so small ion and small ion and some small molecule can communicate to each other. The protein name is connexin. Okay. So last year there was the question: what was the protein name which plays important role in gap junction? The name is connexin. Okay. You can write down Korean. You can write down English. Either way is fine. Okay. So I mentioned that most important thing in the social, in society is communication with each other, okay? Communication, okay? most important thing. Okay. Okay. Communication, most important. What is another thing most important in social society in social work? 사회생활할 때 소통 말고 또 뭐가 또 중요할까요? 좋은 리더가 되려면 어떻게 해야 될까요? 회사 생활할 때 저는 회사에서 부장까지 하다 왔는데 서진이 좋은 부장이 되려면 어떻게 해야 되겠어요? 소통도 잘 돼야 되지만 좋은 부장이 되려면 사람들 이름을 다 기억해줘요. 부장일 때 제가 저 밑에 한 30년 정도 있었거든요. 이제 또 신입사원들이 오면 다섯 여섯 명이 오면 이제 항상 와가지고 이제 인사를 해요. 이제 누구라고. 그 당시에 최대한 이름을 기억하려고 노력합니다. 아니 이제 이름 아침에 까먹었지만 네, 그때는 이제 그냥 인사하고 그래 회사 생활 잘하고 오케이. 다음에 그때 만났을 때 저는 꼭 이름을 불러줍니다. 그래서 그 사람들이 나를 대한 평가가 싹 틀려요. 노력하세요. 노력하면 돼요. 잘 못 외우는데 최대한 씁니다. 지금도 수업 들어오기 전에 계속 다시 한 번 더 혹시나 내가 애들 이름 다 못 외울까 봐서 사진을 프린트해가지고 이름을 계속 보면서 다시 리마인드 하고 와요. 왜? 중요하기 때문에. 사람과 사람과의 관계에서 중요하기 때문에. 커뮤니케이션도 중요하지만 이름도 외워주고 하면 호감도가 싹 달라져 버려서 별 어렵지 않습니다. 노력하면 됩니다. Okay. So we covered connecting. Okay. This is let adjacent cell communicate to each other. Now we will cover this cell membrane also can adhere to extracellular matrix. In chapter 5, we cover extracellular matrix. So outside the cells are surrounded by extracellular matrix, especially the collagen, and they are composed of proteoglycans like this, especially the cell. In the cell, they have integrin protein, like integrin protein. So this protein is bind to the outside extracellular matrix, and also this protein can bind to the inside cell. Integrin is bind to the actin filament in the inside cell, but outside also integrin can bind to the outside of cell matrix is like this. Okay. Okay. Now we will cover the third subject. Substances can cross membrane by passive processes. First one we call it as diffusion. Okay, diffusion. And next we will cover facilitated diffusion, and also we will cover another facilitated diffusion. First one is diffusion. So in the water, when we drop one drop of ink, so 1 minute later, the water becomes like this. Because the ink can diffuse in the water. The diffusion means that process of random movement toward a state of equilibrium. So it means that this particle can move randomly. Finally, they will make the equilibrium like this. Okay? Let me see here. So, figure 6.9 show that permeability of phospholipid bilayer. Okay? This is phospholipid bilayer. For example, small and uncharged, because the phosphorite this inside, they are very hydrophobic. That's why the uncharged molecule pass through the hydrophobic lipids. However, large molecule and some kind of ion and the amino acid, they cannot penetrate the lipid bilayer. Okay? The small uncharged molecule pass through the hydrophobic lipid bilayer. However, large molecule and also the ion Ca2 plus, Na plus, they cannot penetrate the hydrophobic region of the lipid bilayer like this. Okay? So, for example, this kind of CO2 can diffuse lipid bilayer. They are randomly movement to inside, so finally they will make equilibrium. Okay? Now we will also cover osmosis. So osmosis is also diffusion. Osmosis is the diffusion of water across the membrane. Okay, we have to know the isotonic, hypotonic, hypertonic. Okay. Okay, if we put our red blood cell in the isotonic material, the rate of the water movement going out, going inward, they are equal. However, when we put our red blood cell in hypotonic cell, it means that so inside we have lots of the molecules like this. Outside there is not, there is less. So in this case, water is cells take up the water. Take up this water to make equilibrium. So finally the cells take up water and swell and burst. Okay? So when we put cells in hypertonic, okay? So when we put our red blood cell in hypertonic, it means that the water inside cell move to the outside. Water can be outside. Cells lose water and they will make it shrink, so like this. Okay, so osmosis is diffusion of water. Water diffuse across the membrane to make the equilibrium. So next is we will learn the posterior diffusion. This is diffusion, but diffusion that is aided by channel protein. Okay, we also have another postulated diffusion too. Okay, let me see the first one. Okay, so this one is that. So previously we learned that so large molecule or the ion cannot penetrate the phosphobilayer layer. However, our membrane has the channel protein is like this. Okay, so because we have the channel protein, so this kind of the polar substances can pass through the lipid bilayer. Okay, for example, the outside cell, there are lots of the polar molecules. Okay, but they cannot penetrate the phosphobilayer. However, there is a channel protein is there. So some the ligand bind here, they open the channel protein. At that time, for example, the concentration of this substance, this is higher, this is very low. So this molecule can diffuse, okay, to make the equilibrium. So polar substances can pass through the hydrophilic, so the channel protein, they are very hydrophilic, or hydrophilic means that they like the polar, they like the polar molecules. They can pass through like this. So next one is that, so for example, so in this case, okay, several polar molecules, okay, Ca plus and or Na plus, the polar molecule can pass through like this. The next one is that this is also postulated diffusion, but this carrier protein, channel protein, can allow specific substances to cross the membrane. For example, this is the outside cell, and this is inside cell. This is glucose. The concentration of glucose is higher here, very lower here. Okay, but this carrier protein can only bind to the glucose. Okay, that's why the glucose can be passed through this membrane. Okay. Previously, that protein, this channel protein, they don't have specificity, but some kind of the channel protein has a specificity. So they can allow specific substances to cross membrane. Okay. So for example, this is x-axis, glucose concentration of outside cell. So this is diffusion rate. So we will find that sometime at which all available transport are being used, at that time they will make the rate of diffusion did not increase. They will make this kind of the flat region like this. Why? Important. Okay, why? Why this kind of thing is happen? The transport which can transport glucose are occupied by the glucose. It's at higher concentration of glucose concentration, all of this channel protein is occupied by the glucose. That's why the diffusion rate becomes flat, is like this. Okay? Important. Okay? Next one, we will cover the fourth subject, active transport. Okay. Diffusion means that at higher concentration, this is lower concentration, higher concentration and lower concentration. From higher concentration, so also this is a higher concentration and lower concentration. However, active transport move substances against concentration. Against concentration. That's why the energy, ATP energy is used. Okay? The important thing is that move substances against concentration. That's why the ATP energy is used. There are three kinds of the transport. First one is uniporter, symporter, antiporter. For example, uniporter moves one substance in one direction. Symporter is two substances in one direction. Antiporter means that one substance is this location, this orientation, but the other one is this orientation like this, in opposite direction. There are three kinds of active transport. Okay? So, in active transport, there are two kinds. First one is primary active transport. Second one is the secondary active transport. So we will see some details. Okay? Let's start from the primary active transport. Primary active transport, they also need ATP. For example, so this is natrium and this is an Na plus. Okay. So the concentration of Na plus here, they are very low, but they are very higher. Outside they are higher. But this active transport the Na plus against the concentration. Okay? But they use ATP like this. Okay? So this primary active transport, okay, requires direct hydrolysis of the ATP. Okay? So after they use the ATP, so they can move through Na plus to outside. During that time, two K plus can be transported in the inside. It's like this. So one is here, the other is here. So we call it as this is an antiporter. Okay. Okay. So example is a sodium potassium pumper. So they export 3 Na plus ion and brings 2 K plus ion together. The important thing is this channel protein directly used ATP. Second one, we call it as secondary active transport. Okay. So actually this is a primary active transport, and this becomes secondary active transport. They do not use the ATP. This one, they directly use ATP. That's why we call it primary. This one becomes secondary. Okay, let me see some details. Secondary active transport. This is the an ion concentration gradient established by primary active transport power the secondary transport. For example, this is the sodium potassium pump. Okay, sodium potassium pump. They pumping out Na plus. Okay, so it means that concentration of Na plus, the outside that is very high. So the ion concentration, especially here, Na plus concentration, this is higher. Inside is very low. So Na plus can diffuse by using this secondary transport. So at that time, actually for example, glucose, outside the cell, glucose concentration is very low. However, the inside cell, glucose concentration is very high. But this secondary active transport the glucose against the concentration of glucose. Okay? So ion concentration established by active primary transport powers. This is import. This is import. They transport same two protein together, two things together. At that time, this glucose can be moved against the concentration. Okay, so this is example simple drives the transport of glucose against its concentration. But they did not use the ATP. Okay. Now we will cover the final subject, large molecule. Okay. For example, glucose and some amino acid can be transported by using the channel protein. They are okay. However, some kind of the bacteria, so when glucose size is like this, bacteria is very big, is like this. So some kind of channel protein cannot transport the bacteria. So at that time, we used endocytosis or exocytosis. Some very big molecule, some kind of the proteins. They are composed of several amino acids and also polysaccharide and some kind of nucleic acid or some kind of bacteria. So they are very large to cross the membrane. At that time, they can be taken in or excreted by means of receptor. For example, we call it as endocytosis. Okay, for example, solid particle. So, for example, the membrane, it pores inward and forms receptor. Some kind of extracellular fluid, they can make the receptor like this. Okay, the main difference between the phagocytosis, pinocytosis, phagocytosis, they made a pseudoproteome like this. For pinocytosis, they did not make the pseudoproteome. So phagocytosis means that they can uptake non-specific uptake of large molecules like this. Okay? Pinocytosis means that non-specific uptake of extracellular fluid. Okay? 이장은 알겠어? 이해했어? 친구들 언어머리나. Very important. Okay, the difference between phagocytosis, pinocytosis. Okay, phagocytosis means that non-specific uptake of large, undissolved particle. Pinocytosis means that non-specific. Okay, star and scale, they can be go inside extracellular fluid. Final one is receptor-mediated endocytosis. This is specific. For example, they can only transport stars like this. Okay? So they have the receptor protein. So receptor protein in coated pit, they can bind to the specific proteins. So they will make the coated vesicles like this. The large molecule but specific molecule can bind to the receptor. So they will make the vesicles like this. This is recorded as a receptor-mediated endocytosis. Important thing is they can bind to the specific macromolecules. Okay. Final one is the exocytosis. Okay. So sometimes we have lots of the undigested material inside the cell. At that time, this large molecule cannot penetrate this membrane, or even though there is the channel protein, they cannot be transported. At that time, we used exocytosis. So vesicle membrane fuse with cell membrane, but the material packed in the vesicle, sometimes the digested enzyme or lots of the neurotransmitter can be secreted to the cells like this. This is recorded as an exocytosis. So table 6.2 summarizes the endocytosis and exocytosis. Okay, so exocytosis is composed of three: receptor-mediated and pino-carbo. Carbo means that non-specific, large molecule. Pino, non-specific, extracellular fluid. Receptor, specific. Okay. In exocytosis, they can release large molecule, and also they can release small molecules. Okay. So today we finish it. Chapter 6. Cell membrane. Did you have any question in cell membrane? Okay. 다 이해했나요? 시험 맞춰 보셨나요? Okay. Marine and Rhea, did you guys understand it? Okay. Okay.